Record. Definitely. Definitely I was gonna forget because my mind puts medicine in my head and everything else just kind of leaks out. So, all right, dysentery. We have Campylobacter, right? Salmonella, Shigella, EHEC. So EHEC, remember, what can EHEC cause? Who remembers? What is EHEC causing? The problem with EHEC. Does anybody know? This is the one that causes HUS, correct? HUS. Remember, we talked about HUS. HUS, we said, rat, three things basically: renal failure, renal failure. It has anemia, special type of anemia, right? MAHA, microangiopathic hemolytic anemia. You also have thrombocytopenia, thrombocytopenia. Okay, now also, many people don't know, but also, if you look at E. coli in general, E. coli is a gram negative, right? So try to remember, gram negatives tend to have, so let's say this is a gram negative, it tends to have a PAMP. This PAMP, we call this a pathogen associated molecular protein. It's LPS. That's the LPS. Gram negatives have LPS as their PAMP. Macrophages are going to have toll-like receptor 4. This will bind to PAMP. When these two things bind together, you're going to release something called NFK beta. So why am I telling you this? NFK beta is an immune modulator. NFK beta is responsible for releasing certain types of cytokines. So let's take a look at that. Certain types of cytokines like tumor necrosis factor alpha, IL-1. You guys can remember that? IL-2. So let me put you this list right here so you guys can all make sense of it. And let me give you a nice little drawing because my drawings are not the best. Let's take a look at this. Right? Macrophages engaging with the gram negative. After this reaction happens, you tend to release a lot of NFK beta. NFK beta is an immune modulator that releases these type of cytokines. This is what happens. So technically speaking, if we look at E. coli, we mentioned it can cause HUS, right? Especially that strain, EHEC can cause this. But we should also be very clear because if you have lipid A getting released, lipid A can activate a lot of different pro-inflammatory factors. For example, lipid A, it can actually activate something called tissue factor. And you remember, tissue factor can actually stimulate the extrinsic pathway, right? In coagulation, leading to coagulation cascade activation. So we can have a DIC with activation of lipid A. So technically speaking, E. coli can cause a HUS and a DIC. Does this make sense? Because lipid A can trigger this type of immunogenic reaction with tissue factor. Good. All right, that's good. So yes, we talked a lot about E. coli. E. coli has a whole bunch of different types of toxins, a lot of them. We went through all of them in our class. Yes. So basically, LPS is another one. Let's add LPS to the list. Verotoxin is one. LPS is another one. If we have activation of LPS, we just basically said LPS can lead to DIC, but worst of all, it can lead to a septic shock. Because if you activate tumor necrosis factor alpha, this can lead to a septic shock. So we can add that to the list. Septic shock. Okay, good. Now, remember that E. coli is also encapsulated. E. coli is an encapsulated organism, right? E. coli is encapsulated, so it can tend to cause things like meningitis. It can get its way into the brain. So remember, for neonatal meningitis, the most three common pathogens, we have group B strep, we have E. coli, and we have listeria. That makes sense, right? So E. coli is number 2. the most common type of pathogens for neonatal meningitis, also for neonatal sepsis, right? This makes sense. Now, just to ask you a question, if you have a maternal, let's say 35, 37 weeks gestational age, you check for group B strep to see if she's positive, because group B strep is the most common cause of neonatal sepsis and meningitis. Let's say she is positive. What will we give her? We can give her a type of drug. What drug can we give her? Let's see, who knows? What drug will we be giving this patient who's positive? Penicillin is correct. Penicillin is correct. Yes. Yes. So now that we mentioned penicillin, what is the mechanism of action of penicillin? How does penicillin actually work? Penicillin is a beta lactam drug, right? We all agree there. Penicillin. So how does penicillin actually work? What is the mechanism of action? Inhibits cross-linking. Yes, it does inhibit cross-linking. And it does this, yeah, it inhibits cell wall synthesis. That's good. So all these answers are correct. If you think about it, so if I were to write this in a complete sentence, I would have to say it inhibits cell wall synthesis by blocking peptidoglycan cross-linking. So cross-linking can occur, right, on the cell wall. What enzyme is responsible for this? Transpeptidase. Transpeptidase is a type of PBP. Transpeptidase is the active version of PBP. So in a complete sentence, penicillin inhibits cell wall synthesis by blocking peptidoglycan cross-linking, by inhibiting transpeptidase. That's like the complete sentence there, right? And of course, transpeptidase is a PBP, penicillin binding protein. That makes sense. Now, there's another mechanism as well that we need to know. It's also on the first aid. This mechanism is the activation of autolytic enzymes. So whenever this wall is weak, these prokaryotic cells tend to activate autolytic enzymes. Autolytic enzymes. In order to destroy the wall so they can replenish it. That's one thing that it does. So let's recognize that as well. So that's another mechanism as well. Okay, something else that we should also know, something else that's not everywhere, but we need to know this. These NAM and NAGs, look how they stack up horizontally. The process of linking them at a horizontal level is called transglycosidation, done by the enzyme transglycosidase. It's able to link them horizontally, like this, creating these groups. Okay? So this area right here, where they're able to bind to one another, this area is a D-ala area. D-ala. This is where vancomycin is having its mechanism of action. This is how vancomycin works. It's blocking this area, not allowing for the transglycosidation to occur. So you can't stack your glycoaminoglycans. You cannot stack these N-acetylmuronic acid and N-acetylglutamic acid. They cannot stack up. That's what vancomycin is doing. Does that make sense, everyone? Does that make sense? So this vancomycin, yeah, do not confuse that with penicillin, two completely different types of drugs here, right? So what are the side effects of vancomycin? Who can tell me some of the side effects of vancomycin? Yes. Red man syndrome, good. Red man syndrome, yes. So I just remember NOT. I remember it has nephrotic issues. It causes ototoxicity, ototox, and it also causes thrombophlebitis. But then it has that weird symptom, of basic red man syndrome. Red man syndrome, yes. Good. It's actually going to degranulate the mast cell. And it doesn't actually bind to the IgE. It has nothing to do with the IgE binding. So let's get it straight. This is a non antibody stimulus of the mast cells. Basically going to go in and stimulate a second messenger system of the mast cell, allowing for histamine to be released. right? And remember, mast cells also release trip tryptase, which is going to be this basic marker for every time you have a degranulation or a mast cell degranulating, you have a tryptase present as well. Does that make sense? Good, good, good. Let's continue. Let's continue. All right. So yes, that's how we're looking at it. Penicillin makes sense to me. Good. So yeah, we did mention. So okay. So far, we mentioned E. coli. We mentioned different types of strand. Now, E. coli, we also have to remember, it can cause diarrhea. It can cause this type of watery diarrhea. So you remember, does it cause secretory or osmotic diarrhea? What type? Who remembers? And we know the difference between them at this point, right? We understand the difference between them. Secretory, right? Yes, secretory. We know this because it's has to do with the activation. So you have heat libel type, and heat stable. Heat stable. You remember these different types of toxins. Heat libel versus heat stable. You have stable on the ground. This is the GMP one. This one is going to activate guanylyl cyclase. While you had the AMP, the cyclic AMP, and this is going to activate adenylate cyclase. So remember that. You have GS proteins, which will activate adenylate cyclase. Adenylate cyclase turns ATP into an increase of cyclic AMP, right? Cyclic AMP, whenever that's increased, it's going to activate protein kinase A. Protein kinase A is able to phosphorylate certain types of proteins at an intracellular level. And that phosphorylation has to do with channels. There's a channel here which will permit the efflux of chloride. So chloride is going to spill on to the GI lumen, chloride. And guess what follows chloride? Sodium. Chloride spills out, sodium follows, and that brings with it H2O. So you end up having a whole bunch of H2O increasing in the GI lumen. This is secretory diarrhea. Secretory diarrhea. As opposed to osmotic diarrhea. Remember, the mechanism for osmotic diarrhea is completely different. Typically in osmotic diarrhea, you have some kind of osmol, something like, let's say, glucose that's not properly broken down, either because of some kind of blunting or atrophy of the villi, or because basically, let's say, I can give you an example. Basically, let's say you have a disaccharide or some kind of drug. That's what I was going to say, some kind of drug. Like, for example, alpha glycosidase inhibitors. In diabetes, there's drugs like Migatol. There's drugs like Migatol and acarbose, which will inhibit alpha glycosidase, not allowing you to break down your disaccharides into monosaccharides. So if you have disaccharides that are found within the GI lumen, is going to have a stronger osmotic pull, which will pull H2O into the lumen. That is what we call a osmotic type of diarrhea. That is the osmotic type of diarrhea. Drugs, yes, lactose intolerance as well, because if you cannot break down lactose into galactose and glucose, you will have a very strong, let's say lactose has a very powerful osmolality. It's going to pull with it H2O. And that would increase H2O in the lumen of the GI, leading to osmotic type of diarrhea. That's the difference. Let's understand the mechanism of differences of diarrhea. Super important to see it that way, right? Okay, good. Also, it can happen with any type of issues with malabsorption. Anytime you have some kind of blunting of the brush border enzymes. Brush border enzymes, you need them in order to break down all these type of disaccharides, right? Into monosaccharides. Remember that. Good. Let's continue. So, okay, basically mentioning E. coli and how it causes diarrhea. We basically mentioned almost all the toxins, all the virulence factors. One virulence factor as well that it has to do with is it's P. fimbriae. P. fimbriae. All right, so this is basically used to travel. It attaches to the uroepithelium and it travels up the urinary tract. So definitely can lead to issues with cystitis, and it can make its way up even higher and cause pyelonephritis, right? It can make its way all the way up to the kidney and cause a pyelonephritis. If patient has a pyelonephritis, remember, we are afraid of a complication where he can either have an abscess or some kind of dissemination, some kind of E. coli in the blood. That's bacteremia. E. coli in the blood, bacteremia. If it activates in the blood, septicemia. That's what we are avoiding at all costs. That's why it's so dangerous, right? Keep that in mind. Keep that in mind. Let's keep flowing. Yes, yes, yes. Okay. So let's see what else. So in this case, that's basically E. coli. We were mentioning before, we mentioned, let's see, we mentioned different types of disease microorganisms, group B strep, E. coli, listeria. So who remembers listeria? What is listeria? Is it gram positive or gram negative? Who knows the profile of listeria? Is related to baby infections, says Roriana Gonzalez. What virulence factor is associated with baby infections? This is going to be the same type of virulence factor that is involved in neonatal meningitis. This is going to be the K1 capsule. That's what allows the neonatal meningitis. That's what allows the travel of E. coli onto the meninges. That's the problem. When they have capsules, they're able to travel. So, yes, definitely. Gram positive says for listeria. Yeah, listeria. Definitely going to be a gram positive type of issue. What kind of medication do we give for listeria? Who remembers? Listeria, what's the treatment? Ampicillin, ampicillin. And mechanism of action of ampicillin, we have already mentioned. Mechanism of action, what was it? Let's see. Ampicillin is first line, definitely. Cell wall inhibitor. Yes, good. Now, I am going to remind you one thing that's very high yield about these kind of beta lactam drugs. You can give them in combination with aminoglycosides because they have synergistic effect. So if you think about it, one of them is working on the cell wall while the other one is working in the internal ribosome, right? They're internalizing the mechanism of action. That is aminoglycosides. So remember, who remembers the mechanism of action of aminoglycosides? Who can tell me that? Aminoglycosides, 30S inhibitor. So let's remember that. Let's remember the 30S inhibitor. So let's look at this. Let's say we have the initiation complex. The initiation complex is made up of different types of, let's say, pieces, right? We have the 30S, we have the 50S, we have mRNA, and we have tRNA. So these elements are going to make up the initiation complex. Whenever you want to have protein synthesis, you want to have translation. Remember, transcription is you creating your mRNA. Translation is you taking the same RNA and making proteins. That's going to be basically transcription versus translation. Remember that. So these are the elements that you need for translation. So how does this happen? How does this start? Well, let's look at that. Let's pay attention here and let's get this nice and neat. So let's say mRNA leaves the nucleus. Boom, mRNA, right? Remember, for eukaryotic cells, we have to undergo some specific type of process here. mRNA, we have to do CPS, Child Protective Service. We have to do capping in this order. Capping, poly-A tail. We add the poly-A tail to it. And we have to do splicing. We have spliceosomes. Remember, specific types of SNRPs, which are going to be involved in splicing, right? Spliceosomes. We have to do this as eukaryotic cells. Prokaryotic cells don't have to do this function. So they're able to make their mRNA and put it out in the cytoplasm without that function, without that child protective service, that capping, poly-A tail, and splicing. We, on the other hand, do. So prokaryotic cells, they release the mRNA. Perfect. mRNA has a sequence to it. If you look at mRNA, it has a small little sequence to it, which they call the Shine-Dogonal sequence. It has a small little sequence to it. Let's call it SD, Shine-Dogonal sequence. This Shine-Dogonal will find the 16S part of the 30S. It'll find this part, and it's complementary to it. So 16S has complementary nucleotides to Shine-Dogonal. That's the first step. We're going step by step. And this is the actual step that aminoglycoside will inhibit. Let's understand how aminoglycoside works. It will inhibit the formation of the initiation complex by binding, by blocking. It will bind that 16S part of the 30S, not allowing mRNA to make its way into the 30S. That's the initial, that's the first step. So remember, aminoglycoside has the first letter of the alphabet, the initial letter of the alphabet. Therefore, aminoglycosides block the initiation complex, the first step of translation. Okay? So let's understand that. That's the first step. Perfect. So mRNA is there. mRNA, boom, joins the 30S. Good. And now what happens next? Well, you have intrinsic factors. Usually GTP will react to them. Then after that, you'll have AUG. You have expression of the start codon. So your tRNA now has to charge the specific amino acid that's here on the P site. We have APE. This is just like a little review for you guys. This is something that you guys should have already seen in biochem, in everything, really. You think about it, even in micro. So, okay, APE. So P is being expressed right here. So guess what? The RNA brings in, down here you have the anticodon, which is complementary to the start codon. And if we look at tRNA, let's look at tRNA. tRNA is going to have a D arm and a T arm. T arm is what recognizes the ribosome. D arm is what recognizes the enzyme that charges the amino acid. It's called amino acyl synthetase. Amino acyl tRNA synthetase. This will charge your amino acid. Make sure it's charged with ATP. Remember, it uses ATP. Amino acid uses ATP. Intrinsic factors use GTP. So let's get that straight. Factors, GTP, amino acids, ATP. For some reason, that's all over the books. Very important, I guess. So yes, once you start to show this AUG, the tRNA can now make its way here and bind. Once that happens, you have your tRNA. Then you have the final move, your final step, which is your 50S. 50S comes in and binds this as well. Good. Just showing you basically how initiation occurs. This is the idea of initiation. Initiation, whenever we look at translation. Remember, translation has a couple steps. It has initiation, elongation, and it also has termination. So right there, I was just giving you an example of initiation. So good. Let's keep it flowing. That's initiation. Very simple if you put it in that way. It's just getting all the pieces together. Okay? And remember, there's different drugs that work at different levels. If you want to stop the tRNA from joining the party, you want to stop tRNA from making its way, let's say, what drug will not allow the tRNA to integrate itself into the ribosome 30S? Who knows? So let's say here you have a tRNA already with a charged, and you want to get your next one in there. Process of elongation. You're trying to get that next, the process of elongating. You want to get your next tRNA. I'm sorry, I'm trying it with the E right here. No, backwards. A comes here. E is here. So let me tell you guys then. The drug that would not allow the tRNA. Yes, tetracycline is correct. Yes, you're right. Don't second guess yourself. You're right. It's tetracycline. Tetracycline, basically. If you look up tetracycline, especially on your first aid, you'll see that it'll tell you that it blocks the amino acyl tRNA, basically from joining this whole protein synthesis part. So yes, you are right. Tetracycline. And you can remember that because T for tetracycline, blocking tRNA. It's that easy. A for aminoglycoside, blocking the initiation complex, which is the first step, initiation. Right? And then, of course, while we have elongation, while all that is happening, you have macrolides. Macrolides can bind the 23 part of the 50S. Hopefully, this is not giving anybody any headaches. This is just things that we would have to go through, definitely. And I'll go through this whenever I do micro. Thinking about doing like a intense micro camp where we do a couple days. We'll see how that works. But for now, 50S, I'm sorry, 50 had the 23S, and macrolides can bind here, right? They can inhibit this. That makes sense. So what is the side effects of macrolides? What can macrolides cause? What's the problem with macrolides? Why is macrolides such an issue? What's the side effects? Who knows? Macro. Okay, we're getting somewhere. Hold on. Macro. Increased GI motility. That's why they kind of give it for gastroparesis. Remember, they sometimes give this for gastroparesis. Generally speaking, medication that gets given for gastroparesis, what's like the first line? What's the one that usually gets given for gastroparesis? Who knows? What's a good medication to know when it comes to metoclopramide? Correct. Good. Yes, that is right. Arrhythmia, someone has said it. QT prolongation, definitely. QT prolongation, that is true for arrhythmias. That's a torsades, right? And how do we manage a torsades? Let's say a patient comes in with a polymorphic type of ventricular type of arrhythmia. Magnesium, right? Magnesium if the patient is hemodynamically stable. What if he is unstable? What do we do? Well, you got to stop the medication, of course. But what if and afterwards you have to manage. So what if the patient is hemodynamically unstable? What do we do with torsades? Defibrillation is correct. Yes, defibrillation is correct. So torsades, hemodynamically stable. That's the first thing you have to check. See if his systolic blood pressure is less than 90. If it is, then you have to do defibrillation. Remember that, all right? Okay, good. You have cholestasis. This has rash and eosinophilia. That's the macrolide type of issue. What other drug, what other antibiotic can cause torsades point? Who knows? What other antibiotic causes torsades? That was a good one, George. Good one. Quinolones is correct. Fluoquinolones. That is correct. Now that you mentioned fluoquinolones, what is the mechanism of actions of quinolones? How does that work? How does fluoquinolones actually work? Topoisomerase inhibitor. Yes, good. Also known as DNA gyrase. Yes. So here we have topoisomerase. Remember, you have those supercoils. While you're synthesizing your DNA, you have formation of supercoils, right? Look at that. If we look at that synthesis is going on this way, let's say, it's continuously making it tighter and tighter. So you need to basically cut it, relax the coil, and then snip it back together. So you have topoisomerase 2 and 4. What's the difference? Well, after you synthesize your DNA, you'll look at them and they kind of look like this, like two rings attached to each other. Because why? Because prokaryotic cells have circular genetic material. They look circular. We have helical. So topoisomerase 4 is able to separate mother and daughter strand. They're able to separate them. So that way, they would be like this, separated. Okay, that's topoisomerase 4. Topoisomerase 2 is causing the relaxation of the supercoils. All right, good. And that is true. Remember, It can cause torsades to point. I always say, what are the three side effects? Most common is GI upset, also known as diarrhea. Most common. The fatal is the torsades that we just mentioned. Torsades. And then we mentioned the weird one. There's a weird side effect that happens. It's just a weird one with all the antibiotics. All antibiotics have a weird side effect that's just out of line. It's not really too common, but it happens. Or this one, for quinolones, who remembers? It's a very specific type of side effect. Who knows? Tendon rupture is correct. Tendon rupture in the older population, older and very, very young population as well. Tendon rupture is what they can get, right? It is rare. Yes, it is, but it's worth mentioning it. Definitely. So remember, I like to mention those rare ones just because those things can pop up in exams. It's good to know them. It's good to know them. It's not really like clinically like something that you'll see in a clinic or something, but just to have the idea. For example, with aminoglycosides, they tend to contraindicate. They kind of contraindicated in patients who have neurodegenerative kind of disorders because they can decrease the respiratory rate. Autonomic system, when it comes to respiratory rate, gets inhibited. I know it's super weird. It is, but you can have a hypoventilation. So, and remember, in these type of hypoventilations, we understand that these are restrictive type of disorders that actually have a normal AA gradient. The gradient will be normal. It's a hypoventilation. Hypoventilation tends to have an increase in PCO2. This is aminoglycoside. Remember, you can't give this with Guillain-Barré, for example, for that reason. Those are the weird side effects I'm giving you. Linozolid. Linozolid. What's the mechanism of action of linozolid? Who remembers? Mechanism of action of linozolid, if you know it, type it in. If you know it, type it in. 50S. Yes. 50S inhibitor, but let me tell you how. Let me tell you how. Since we just learned all about the 50S, we just learned all about the 50S. Let me tell you the difference between linozolid and macrolides. Let me tell you the difference between the two. Remember, we talked about the assembly of the initiation complex? We talked about everything coming together. We said, oh, yeah, mRNA goes in with the 30S, right? The Shine-Dogonal sequence matches the 16S. They have complementary strands. They join together. Then we mentioned the next step, which was that the 30S starts to express the start codon. We said the tRNA gets charged, makes its way here. We mentioned that. And then the last thing we mentioned is that once that happens, the tRNA is here, then the 50S joins the party. The 50S comes in, joins the party. And that's the end of initiation. That's how initiation complex happens. Well, guess what? Linozolid binds the 50S at the 23 portion. Here in this stage, it starts to bind at the stage of initiation. Initiation. Macrolides bind at the stage of elongation. So macrolides block translocation. Block translocation. They're binding to the same spot though. They're binding the 50S. They're binding the 50S, the 23 part of the 50S. Both of them are binding it. Just one of them is doing it as they assemble the initiation complex. The other one is doing it as they're doing elongation. So that's the difference between macrolides and linozolid. So linozolid, what's the weird side effect of linozolid? Who remembers? Who remembers the weird side effect of linozolid? Who knows? Type it in if you know. Thrombocytopenia, serotonin syndrome. Good, good, good. Serotonin syndrome is the weird one because it does have some kind of mal inhibiting effect. Monoamine oxidase. Remember the monoamines. Remember things like dopamine, right? These are monoamines. They need to be broken down either by COMT or by MAO. So yes, they're MAO inhibitors. And if you inhibit the MAO, you would have less breakdown of these monoamines. So the monoamines start to build up. And once they build up, they can cause serotonin syndrome. Monoamines are things like serotonin, dopamine, norepinephrine. Okay. Remember that. Remember that. All right. Good, 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 good. So far, so good. We're flowing. We're making sense of it all. We're catching everything. We mentioned some weird side effects. There's a whole bunch of weird ones, of course. There's always weird side effects. Who knows the mechanism of action of metronidazole? Who remembers that one? That one does have some crazy side effects as well. Metronidazole. Who can explain that? Inhibits folate, somebody says, release of ROS species. Good, good. I'm with the ROS species. So, Jessica, let me show you. Let me show you the folate inhibitors. Let's talk about that. Let's talk about the folate inhibitors just so we can understand folate inhibitors, and then we'll get back right back into metronidazole just so we can understand everything. So, dihydrofolate inhibitor was something else, and we're going to talk about that. We're going to make sense of it right now. So let's remember this super important pathway that somehow, for some reason, the first aid only has like one little page on. Yet this is like a huge part of this exam. Huge part of this exam. There's so many drugs here. There's so many different actions and everything. So we'll talk about this right now. Let's take a look. This here is the de novo pathway, right? De novo. De novo. I have to upload the other video that we did as well on that question day. We did a whole bunch of stuff on biochem for some reason. People asked me about biochem, so I just started talking about biochem and it turned into a whole lecture. But okay, de novo. In the de novo pathway, remember, you're either making your pyrimidines or you're making your purines. So something I will tell you guys right now, something just a rule of thumb. If I'm messing with the pyrimidines, if I'm messing with this pathway right here, I'm messing with a lot of RBC type of formation, right? I'm basically disrupting, messing with that pathway, RBC-ish. Okay, so we're more prone to have RBC problems on pyrimidine issues, on pyrimidine inhibition. Purine, I'm looking more at lymphocytic type of cells. I'm looking more at T cells. I'm looking more at immunogenic type of reaction, like decreasing the proliferation of these kind of cells. Does that make sense? Just letting you know, this is more immuno. This is more heme. You can look at it this way, okay? If I have an issue with the purines, I expect to have some kind of immuno cell problem. If I have an issue with pyrimidines, I expect to have more of a heme kind of issue. Okay, that's just something off the bat, just so you could recognize. Now, let's look at it like this. We're seeing here this very important step where uridine is going to try to become thymidine, right? Thymidine. This is that step, that methylation step that has to occur here. So the methylation process is brought to you by B12. And here you're seeing the replenishment of folate. Replenishment of folate goes from tetrahydra to dihydro. And it continuously makes this cycle, it's perpetual cycle. You're continuously replenishing your folate. So here's where folate is very important. Folate. Folate. And yeah, I can send you guys an image to make sense of it if it doesn't make sense, because these are two different things. Very important. Can cause problems though. So I'm going to show you this image. And I'll send this image to you guys as well. Let me show you. Here. Here, I see that. Look how B12 is involved with this pathway here, tetrahydrofolate. And then you're seeing how folate keeps on replenishing, keeps on replenishing. I'll put this on the chat. You guys can take a look at that. But okay, coming back to this. Let's not lose ourselves. All I'm trying to tell you is this. This dihydrofolate reductase, there's different drugs that can inhibit this. So let's get into it. And let's make sense of this. If it's on a human cell, if it's on a eukaryotic human, what type of drug inhibits this receptor? Who knows? This receptor, this enzyme. Who can tell me? Who knows? Methotrexate. Perfect. Methotrexate for humans. Methotrexate for humans. Okay. How about prokaryotic cells? Prokaryotes. What drug? Trimethoprim is correct. TMP. Okay. What about Parasitic, for example, and toxo. What about toxo? Toxoplasmosis. Pyrimethamine. Good, good, good. Pyrimethamine. Yes. Everybody's good on that. That's what I like. Yes, yes, definitely. I'm exposing this. Everybody's knowing it now. That's what I like. Okay, then I also mentioned an honorable mention I'm going to throw out there. Not super high yield, but proguanyl. Some people wanted to know how proguanyl work. How it works is how it works. Proguanyl. It inhibits dihydrofolate reductase, and this is for malaria. Malaria. Okay? Malaria. Now, I gave everybody a nice way to diagnose malaria before. The whole idea of plasmodium. I told prokaryotes, well, prokaryotes, these are just bacterial cells. Bacterial cells, basically. Oh, like which? Okay, so you could treat, yeah, bacteria. You could treat like E. coli, for example. If E. coli is causing you a UTI, TMPSMX can be given. Yeah, things like that. Definitely. Definitely. Okay, so let's look at this. Malaria. How we diagnose malaria? Pretty easy. I'll give you guys a little trick. A lot of it has to do with this fever cycles, but everybody forgets their fever. It's like, oh, when does that fever happen? How do I remember this? Well, here's a little trick. If it's Vivax, okay, so first off, if it's Plasmodium malariae, it has 3 points here. 1 point, 2 points, 3 points. Every point is a day. So I can say every 72 hours, the fever will have this cycle. That's for malariae. Now, Vivax and Oval, it has 2 points. 2 points. And Oval is just a cousin that hangs out with Vivax. In Spanish, we call prima or primo. So I remember that there's 2 points. So we say 42 hours. It's 2 days. And since that's the prima, we treat Vivax and Oval with Primaquine as well. You have to add Primaquine to the management because Vivax and Oval actually have hypnozoites, which will stay dormant at the liver. So you have to treat with Primaquine as well. Okay. And then, of course, you have falciparum, which is with the F. Falciparum does not care. It does fever whenever it wants to. It messes with you. It's very fatal. Falciparum for fatal. Right? Does everybody understand that? Does that make sense? That's an easy way to remember your fever cycles when it comes to Plasmodium. Good, 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 good. Keep that in your mind. Don't let that go. There's so many little tricks in this exam. We're going to go over them so we can understand them. Okay? That's how you get your malaria right away without having to memorize and practice. Just simplify it. Okay? All right, we continue. So, okay, yes, this is the dihydrofolate reductase type of inhibitors, right? We understand that. But we should also mention some other type of stuff as well. There's something called PABA. Remember that. You have PABA and it's called pteridine. PABA and pteridine. I always forget this name. PABA and pteridine. Both of them unite to become dihydroxypteroic acid. Dihydroxypteroic acid. Okay? And we could inhibit this by inhibiting this enzyme. The enzyme that it has here is called dihydroxypteroic synthase. Dihydroxypteroate synthase. This is inhibited by what drug? Who remembers? SMX is correct. These are sulfonamides. Sulfonamides are inhibiting the PABA conversion and pteridine from becoming dihydroxypteroic acid. You want to stop dihydroxypteroic acid from forming. So you can stop this by giving SMX. Now, there's another drug that, technically speaking, is not a sulfonamide. It's not a sulfonamide, but it does the same mechanism of action. It's called dapsone, correct? And we give dapsone for what particular type of treatment? Remember, it's a very particular type of thing we give dapsone for. Let's see if you remember. It says lepra. Okay, I like that. Leprosy, yes. Although it's not the one I was thinking of, but yes. Leprosy, remember, you can also give rifampin. Not TB. not TV, but I'll tell you what. There's patients who have um they have celiac disease. When they develop celiac disease, they get herpetiformis. They get herpetiformis type of ulcers that start to form. This look like vesicles. Looks, yeah, dermatitis herpetiformis. There you go. I'm saying herpetiformis. I'm like, here comes the first name to it. Dermatitis herpetiformis. Yes, it starts to form these type of vesicles, and you give dapsone in order to ensure that you don't have any kind of complications with any kind of secondary type of infection in that sense. Have anybody heard of that before? Is anybody aware of that? That's why we give dapsone for. Yes. So dapsone does have that weird particular type of treatment. So yeah, now you know dapsone. Yes, and also lepra. That was a good one. Remember, lepra. Yes. You also give one other drug for lepra. Who remembers? Remember, we have two different types. For lepra, you have the tubercular form, and you also have the lethal form, the leprematis. Leprematis. And you have the tuberculoid form. It says macro lepromatose. Okay, so let's look at it. Let's look at lepra. Remember, you have the tuberculoid form. This you're going to give rifampin, and dapsone. For the leprematis form, you have to add clofazimine. Same three regimen, but you add clofazimine. Right? Remember this. Tuberculoid form, the tuberculoid form, what does that even mean? That means that it's going to trigger Th1 type of reaction. Th1, while leprematis triggers Th2 type of reaction. Th2 type of reaction. Th2 is a lot more dangerous. So let me open up a new board just because so it won't lag. Giving class all day. Yes, yes, yes. Okay, so let's look at it like this. Tuberculoid form. What happens in the tuberculoid form? Let's go run through it. Let's explain it so it can make sense. Tuberculoid form. Let's say, for example, you have a macrophage or an antigen presenting cell making contact with this lepra, this kind of lepra. So let's put it like this. Okay, and it's causing a reaction where you are now releasing NFK beta. Remember, we mentioned NFK beta releases a lot of different types of cytokines like IL-1. Let's say IL-1, IL-2. Basically releases 6, 8, and 12, and tumor necrosis factor alpha. So remember, IL-1, very, very important when it comes to fever. IL-2, very important when it comes to T cell proliferation. T cell proliferation. Remembers that. Proliferation. Also, IL-6, these are going to trigger acute phase reactants. Remember, acute phase reactants are all the way in the liver. You go to the liver and you activate acute phase reactants like hepcidin. Hepcidin holds on to iron so that way the bacteria can't get its hands on it. It rigs the system. IL-6 rigs the system. It gets our body, since the fight is going on in our territory, we're able to prime our body in order to win the fight, in order to win the immunogenic type of battle. So IL-6 is what triggers acute phase reactants. Now there's a problem as well. Remember, acute phase reactants also includes amyloid A. Amyloid A. So if this acute phase reactant type of reaction goes on for long enough, that's why it's called acute, because it's not supposed to go on for very long. But if it does, it's going to have some complications. So with hepcidin, the complication will be anemia of chronic disease, right? Hepcidin does increase with IL-6. Yes, definitely. Definitely, Jessica. And what ends up happening is that hepcidin will sequester, is elevated in, yes, an inflammatory disease. Yes, yes, an acute, especially if they're acute, because your acute phase reactants will be around. Now, if this becomes chronic, this is terrible because now you're having hepcidin enforce the hiding of iron, right? The basically hiding the iron from us, and then it causes anemia of chronic disease. That's the complication here. Anemia of chronic disease, right? And then what else? Amyloid A, that's also something that just goes with the mix. As you increase more acute phase reactant happening here in the liver, you start to release more and more amyloid A. Amyloid A starts to deposit itself in our own tissue, causing complications. So let's remember one thing when it comes to amyloid. Proteins, you like to have your proteins in groups of two. You can break them apart, digest them very easily. These are called quaternary types of proteins. Quaternary proteins are things like hemoglobin. That makes sense. You could easily digest them, break them down, reuse them, recycle them, do what you have to do with them. But if you have a tertiary protein, now you're having like a third wheel. You're seeing one that just kind of lingers there. So as you digest one with ease, the other one starts to linger. And this can get phosphorylated. This can undergo some type of reaction and start to linger in our body. That's the problem there with amyloid. So here you have something called amyloid A, which starts to increase in the body. The longer you have IL-6 on the liver, the longer you have amyloid A. That's the problem there. So you can have this type of amyloidosis. They have AA. They'll say A, acute phase reactant. Remember, there's different types of amyloid. There's AL, like light chain. Remember that gamma chains. When you have an immunoglobulin, let's just look at it like this. Immunoglobulin, you have a light chain located right here. This is going to be a light chain. Heavy chain is on the other side. But a light chain, this little piece, again, when you digest everything, this little piece likes to stay, linger, go around your body and create amyloid. So what ends up happening is that you'll have light chains, and that can cause a lot of kidney problems. You have kidney issues in that sense. That makes sense. That's what you see in multiple myeloma. Why do you see light chains in multiple myeloma? Think about it. Our most abundant protein is albumin. Then we have alpha proteins, right? We have beta proteins. And then, of course, we have gamma proteins. Gamma proteins are used to make immunoglobulins. So naturally, this looks like this. Naturally. But when gamma proteins are high, now it looks like an M spike. Kind of looks like an M. I mean, I don't see it. That doesn't look like an M to me. Look at this. This is not an M. But I guess so. So that's why they call the M spike. Yeah, the immunoglobulin is supposed to be properly broken down. But since multiple myeloma is just making a whole bunch of them, and basically we're not able to break them down properly. So what ends up happening is that you then have lingering of this light chain. And this light chain starts to get phosphorylated, starts to react with other things in our body, oxidated, anything. What ends up happening is that it starts to then linger in our body and cause these type of amyloid reactions. That's why we have light chains, right? Light chains. That's called AL, amyloid of light chains, that you see with multiple myeloma. So amyloid is a very interesting topic. Definitely. There's different proteins that do different things. And with Alzheimer's, you're going to have as well different types of tertiary proteins. So it's not difficult. If you're understanding what's happening, a lot of it makes sense. But let's get back to the topic. We were talking about the tuberculoid form, and we said that the tuberculoid form starts off when antigen presenting cell recognizes lepra, let's see, the leprous disease, and then we basically have an immunogenic type of response when NFK beta is released. When NFK beta is released, it releases all these cytokines. We got up to acute phase reactants, and I just started going off the top of my head, just talking about IL-6. We now understand IL-6 pretty well. Good. Also, one more thing I want to add to IL-6, don't kill me, but also C-reactive protein. C-reactive protein also increases, and that is more of a marker that lets us know when inflammation is going on. That's also increasing with this. Okay, then we get back to IL-8. IL-8 is chemotactic. IL-8 is chemotactic. It will actually call over the neutrophils. The neutrophils are blind. The neutrophils are navigating in a blind field. They don't know where they're going. They're completely blind. But they need chemotactic factors in order to see where the action is going on. Where is the battle happening? They will utilize these chemotactic factors. So I'm going to give you an updated list of all the chemotactic factors that we can find. And these are the important ones to know. These are the ones that I've seen. These are the ones that could come out in any type of MPME, any exam. This is it, right? So I'm going to put this on the chat as well. You guys can download this. C5A. So my mnemonic was chemo silk. Chemo silk. So chemo was for C5a, and then 5-HPET, IL-8, leukotriene B4, kallikrein. But then again, they added these two. So be careful. They added these two, so be careful. Yeah, got to keep updated all the time. Updated all the time. Okay, let's continue. Then you have IL-12. Remember that IL-12 is able to turn your T helpers into Th1. Th1 reacts with tumor necrosis factor alpha, can release interferon gamma. Interferon gamma will recruit more macrophages. It recruits macrophages. So far, we're understanding everything is making sense. All I'm saying is that when you have the presence of IL-12, you are favoring Th1 production. Remember that IL-12 inhibits the naive T cell from becoming Th2. Remember that inhibits this. And when interferon gamma is released, remember that this also works as a inhibitor of your naive T cell promoting the Th2 type of reaction. Interferon gamma is also involved in this step. Okay? Recognize this. This is basically the tuberculoid type of reaction. This is the granuloma type of reaction. This is what you get. So hopefully everyone knows this. You can definitely practice this in your brain. It's very simple. It's not too crazy. You're recruiting more macrophages. So how does the story end? So this is what happens. Macrophage, like we said, reacts with, let's just say a bacteria, just not to single out lepra. There's a bacteria. And in that case, we said it releases NFK beta. NFK beta stimulates all those different types of cytokines that we said, IL, 1, 2, we said 6, 8, 12. And then we said 12 in particular will allow T cells to become Th1, right? And we said, well, I'll tell you how that can happen as well. Macrophages can actually bind to T cells. Macrophages have MHC2. MHC2 binds CD4 positive cells. And they have a co-stimulatory type of effect where your CD28 will bind to B7, right? And that will lead to the release of cytokines like IL-12, let's say. And that will lead to differentiation, right, of this T cell, activated now to a Th1. Hopefully everybody understands this. This is something very basic for this exam. You have to know these steps. How are you making your Th1 cell? And as this is happening, any kind of naive T cell that's in this area is also going to undergo differentiation because of the influence of IL-12 and the influence of interferon gamma. Okay? Once this happens, now you have Th1. And we said Th1 is going to call upon, is going to utilize interferon gamma to call upon recruitment of more macrophages. So more macrophages will come. So let's see how that looks. More macrophages are now arriving to where there was war, where there was a battle. More macrophages. Here you have more of them arriving. Macrophage, all engaging, all causing the same type of reaction of IL-12, all of them differentiating your naive T cells to Th1, the Th1 calling even more macrophages. So more macrophages are coming. If the macrophage cannot destroy this bacteria, they will continue their proliferation. If they destroy the bacteria, then the incentive for more macrophages will decrease because you won't be making Th1, you won't be making interferon gamma. So if it doesn't, then you have high incentive of macrophages to arrive. Now, once these macrophages all arrive in this one area, yes, granuloma formation. Yes, once they arrive to this one area, their cell walls, let's say their membrane is very similar to one another. So they're pushing, they're shoving, they're pushing and shoving. And what ends up happening is that they end up joining and becoming one cell. So under microscopes, people look at it and they say it's multinucleated giant cells. That's not really the case. They're not giant cells. They just happen to smudge together. They just happen to come together. Multinucleated giant cells. In reality, they're just macrophages that ended up smashing together. So that's why they look like that under the microscope. That's why they have that name. That's why they're giving them this name. Now, if there is a microorganism inside that granuloma, it's going to have caseous type of granuloma. If this reaction is fake, meaning if it's just an autoimmune reaction, if our immune system is just acting up and creating these granulomas without a war, without actually killing a microorganism, then it'll be non-caseating. Non-caseating. For example, in sarcoidosis, you should have non-caseating because there is no culprit in the middle. There is no bacteria that we're trying to kill in the middle. In Crohn's disease, it should be non-caseating. There is no bacteria that's being put up in the middle to try to kill them. Anything that has the bacteria in the middle that we're destroying, that we're fighting up against, is caseous. It's like cheese-like, they call it. That's how I know if it's a real type of actual infection versus my immune system just acting up. Definitely. TB, you tell me. TB is a microorganism. It's in the middle. Caseous. Yes. If it's autoimmune, non-caseating. Like Crohn's, for example. I don't even need to look it up. I just have to know, hey, what are we killing inside that granuloma? Nobody? Then it's non-caseating. If there is yeah, definitely. So don't go out there memorizing. All you have to know is what does it mean? What's going on? Let's understand medicine this way better. That way we don't have to go off memorizing a whole bunch of things. Yeah, definitely. If you are attacking a microorganism, caseating. That should make sense. Yes. Thank you. That's how we do it. You want to understand this. If you understand it, it's less for you to try to memorize. There's some things though that just memorization that there's no way around. But there's anything that we can help, we have to try. And this is something that, yeah, you see the difference there. That's a tuberculoid type of form of reaction. That's the tuberculoid reaction that we're seeing. All right, everybody, let's leave it here. We already have an hour and 22 minutes. I'm going to try to keep the drills an hour or less than an hour because it's an overwhelming amount of information. And we don't want to overload our brain, right? So let's leave it here. So today we were okay. We crept into, we did some micro, but we crept into some pharm. And then we also crept into some immuno. Some immuno got crept into as well. So we were able to check that out. I want to be more consistent with the drills. Definitely, I'm offering drills. Join my Patreon if you're not on my Patreon, because whenever I do these kind of free drill sessions, definitely you can take part in them. I load them up on patreon.com. Definitely, I'll show you the website. I'm putting them in there all the time. I'm going to try to do a lot more drills this month as well. I haven't put everything in the calendar. Definitely, thank you everybody for joining me. This will be recorded, and I will put it for the Patreon. So anybody who joins the Patreon will see it right there. Yes, definitely. Thank you everybody for joining me. I really appreciate it. Definitely appreciate it. And I'll continue with these drills. I'm seeing, yes, it is helping people, so I'm going to keep doing them. I'm going to see if I continue basically a lot of micro and pharm, go that route. Thank you everybody for joining me. Thank you everyone for joining me. Appreciate it. I'll catch everybody next time. And next time, what should we do? We'll start off with micro, and we'll see where it takes us. But there's also a lot of pharmacology. Yeah, heme has a lot of things as well. When we have the whole cascade, that whole reaction, all these drugs that's in it, all these glandsmen. We have so many different things. Biochem, okay, yes, definitely. Microimmunology, I like that. I like that. We'll do everything. I don't mind. Like my brain is just loaded with information. We can just flow for an hour and see where it takes us. Definitely. All right. So thank you everybody for joining me. I appreciate everybody. That's my Patreon. If you're not a member of the Patreon, join the Patreon. Definitely, you'll see more of the class. I'm going to upload this video, and it's free for everybody. So definitely, all members, doesn't matter if you're paying or not, you'll get this video. Okay. Take care, everybody. I appreciate everyone. Have a good night, and I'll catch you tomorrow. We have oncology class. Tomorrow we have a class on oncology. We started a new subject. So remember, I'll see you tomorrow, 8 p.m. Eastern. Oncology begins. Okay. So have a good night. Appreciate everybody. Thank you. Oh yes, I have cardio. Yeah, yes, yes, I have cardio classes. I have everything. I have everything there of what I've done so far. Thank you everyone for joining me. Have a good night. And yes, we'll be doing a lot more drills from now on. Definitely, we'll be doing a lot more. Take care.